Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the LCRS and the DNA binding motifs. Okay, so in case of DNA binding motifs, generally today I explain only the brief term about that, but the types of DNA binding motifs, this is explained by me in the next video. Okay, so you just continue with me. So locus control region. Before going to this topic, I want to say that the factors related to the eukaryotic transcription that are already explained in my channel. So you just check out that all. Okay, this is the locus control region is what? There are cis-acting DNA segments that needed to transcription activation of entire locus or the gene cluster. So cis-acting DNA segments needed for what? For continuing the transcription process or the activation of the entire locus means region or the cluster, gene cluster. So cis-acting region is needed for continuing the transcription process or the activation of the gene cluster. Now this LCRS, that unlike classic enhancer elements and operate in an orientation dependent manner. So LCRS, the main thing is that it helps in the orientation, means that means the initiation process of the transcription. And the LCRS, these are known as the DNase 1, and this is hypersensitive sites. This is hypersensitive sites, and this is known as the locus control region. So main thing is that LCRS, these are characterized by the DNase 1, and the region where this LCRS is present, or it is sequence one kind of, so this is the present where this is the hypersensitive sites. Now LCRS also present in another loci, and including the gene clusters encoding the alpha globin and the MHC protein. So there also these LCRS are found. The gene regulatory proteins that bind the LCR interact through the DNA looping with proteins bound to the promoter of the globin genes. So what is saying that actual activity of the LCRS is totally not understood by the scientist, but general function of the LCRS that is played in the transcription, that is slowly explained by them. And so about the LCRS in the general book, any genetic book, so you can see only the brief part about this LCR, because actual role, what is the role of the LCRS in the transcription process, it is not clearly understood. So they act, how they act, some gene regulatory proteins bind with the LCR. Okay, LCRS, this is the region or the sequence. So some gene regulatory proteins that bind with this, and by how? Through the DNA looping. Okay, through the DNA looping they bind with the LCRS, and with proteins bound to the promoter of the globin genes. So generally this is discovered in case of when this is present in the globin genes. Okay, so from the globin genes some explanation is drawn, and this is I briefly explaining. Okay, so what is saying that this LCR is first bound with the gene regulatory proteins by the process of DNA looping, and then the proteins bound to it with the promoter, and after that promoter when bind to the transcript or the DNA, then the transcription process will start. In this way the proteins bound at the LCR and help attract the chromatin remodeling complexes. And okay, in this chromatin remodeling complexes already have explained in my previous video, so you just check out. So this attract the chromatin remodeling complexes, histone modifying enzymes, and the components of the transcription machinery. So when this LCRS that bound with the gene regulatory proteins, then after that the promoter already has bound with them. After that this complex, the already complex is formed, this complex attract the chromatin remodeling complex. This is one kind of coactivator, and histone modifying enzymes and another components that are involved in the eukaryotic transcription process. So that act in the conjunction with the specific regulatory regions of each individual globin gene, and LCR includes a barrier sequence that prevents the spread of the neighboring heterochromatin into beta globin locus. So here in that way they attract the transcription machinery, and after that the transcription process will be started. Another way, it also prevents the spread of the neighboring heterochromatin and into the beta globin locus. So you just say that the transcription process will be stopped by the presence of some silencer sequences of the LCR's region that prevents the spreading of heterochromatin or the spreading of transcription process in the neighboring gene when in the beta globin locus. Okay, so when this transcription machinery is attracted, all the transcription machinery attracted to the gene, they act the conjunction with the specific regulatory regions of each individual globin genes. That means what is the role of the LCR is, it helps in the conjunction of this total transcription machinery and the regulatory regions of the gene or globin gene. Clear? So its role is, main thing is that it helps in the conjunction process of the transcription. First it attracts the all transcriptional machinery and then make a conjunction process, make a conjunction region with the regulatory regions, specific regulatory regions. Now next thing is that DNA binding motifs. This DNA binding protein, this is another protein, such as transcription factors, this recognize and bind a short nucleotide sequence, usually as a result of extensive complementarity between the surface of protein and the surface surface features of the double helix in the region of the binding. So what is the DNA binding motifs? This is one kind of protein, okay, and the DNA binding protein transcription factor is another protein. This transcription factor, for example, this transcription factor first recognize and bind where a short nucleotide sequence. So first transcription factor recognize a short nucleotide sequence, and as a result of extensive complementarity between the surface, by how they recognize the short nucleotide sequence? Because of the extensive complementarity or the differences between the surface protein and the surface feature of the double helix. So there is a difference between the DNA double helix surface and the surface protein. Okay, the protein that must be bind with the DNA double helix, the surface of that protein, and obviously where they bind of the surface of the DNA double helix, there is a difference. Okay, this difference, due to presence of this difference, this transcription factor first bind with the short nucleotide sequence, clear, of the DNA or of the DNA, and contacts occur between DNA binding protein and edges of base pairs that are exposed in the grooves of the DNA. So now after binding of this, make a contact, okay, a contact is created where between the DNA binding protein and edges of base pairs. Okay, DNA binding protein and edges of base pairs. Because if this binding is not possible, then the contact is not possible between the DNA binding protein and the base pairs groove. Clear? So this is done by these motifs. Okay, this is done by the motifs. And here one important thing is that hydrogen bond provide a sequence specific contacts, and these specific contacts in case of donor and activators that are present in AT base pairs and GC base pair, these are not same. Differences are there in the hydrogen bonding. So this is the role of the DNA binding motifs. Okay. So very briefly I explain here. So this DNA binding motifs is there are many types. So I will try to explain the types in my next video. So continue with my channel. Okay. Thank you.